Maybe Matthew, Paul, Deborah, or teacher Anne. Teacher Anne, do you hear me? Hello, teacher Anne. How are you? Welcome.
Thank you. Good morning.
Good morning. Welcome. Okay, the class for today is science, right? That is the class.
Yes, biology, science.
Biology, science. Okay, very good. Okay, Abby, do you hear me? Matthew, Paul, Deborah, do you hear me? Welcome, guys. The class going to start in a couple of minutes, okay? Actually, yes, in a couple of minutes. We have to wait for more students, and you can start, teacher, okay?
Okay, no problem. I will start in a minute.
Yeah, 2 more minutes, and yeah, in 2 more minutes you can start.
Yes, thank you.
Okay, guys. When teacher is starting in the class, it
Good morning, Professor Anand. Good morning, guys.
And good morning, everyone. Okay.
Good morning. Good morning. Okay, yeah, I would start.
Okay, so we are starting. You're welcome. We are starting today's lecture, and let me share the screen.
Yeah, 1 moment, please. It's ready. It's ready, Professor.
Okay, so today's lecture is the second lecture in the biology series, and we will try to cover 3 topics today. The first one is enzymes. The second one is cell membranes and transport. And the third one is nucleic acids and protein synthesis. So let us start the chapter enzymes. So you guys may have heard a bit about enzymes before, I expect. Do you have any idea, anyone, that what actually enzymes are? You guys can raise your hand, or you can reply in chat, or you can ask for permission, and you can reply verbally as well with your mic. Anyone, what are enzymes? Do you have any idea?
If I am good, are protein that produce chemical change in the body.
Okay, thank you very much. Yes, somehow they do that. Okay, so let us see what actually enzymes are. So enzyme is a biological catalyst, okay? So what is basically a catalyst? Catalyst is anything which speeds up a reaction. Catalyst, like we say that something is being catalyzed, so something is being speed up, okay? So for example, if you want to travel between point A and B, so there can be like many different things that you can try, like you can walk between the 2 points or between the 2 places, you can take a bike. So a motorbike will increase the speed that it will help you to reach point B more quickly, okay? Or you can take a car, or if the distance is far, you can take an aeroplane. So the more you go higher, the more like quickly you are reaching to the point B, okay? So similarly, enzymes work in our bodies and they catalyze chemical reactions. So otherwise, they would take many days or sometimes months or years to complete. So enzymes catalyze chemical reactions and they enable them to complete more quickly. Is it clear? Okay, so, okay, thank you. So let's proceed and let us look at what enzymes are made of. So in our last lecture, we talked about the biological molecules, that we have different types of molecules in our body, but the 4 main types are like carbohydrates, fats, lipids, and proteins. So most of our body is composed of these 4 biological molecules. So let me write it again for you guys. So we have carbohydrates, we have proteins, and the third one is we have lipids, and the fourth one is a kind of fats, okay? So enzymes are made up of proteins, and enzymes are globular proteins. So what do we mean by globular? Just like a globe, okay? So this is, this represents a globular shape, okay? Like a globe. So enzymes are made up of proteins, and what do? They catalyze biochemical or biological reactions in living things. So just like I said, almost all metabolic reactions which take place in living organisms are catalyzed by enzymes. You guys should know what catalyze mean. Enzymes are therefore essential for life. Without enzymes, life is impossible. Okay, we cannot imagine life without enzymes. Otherwise, the reactions that we need would take years to complete, and we cannot wait that long. So many enzymes. So the fourth point in enzymes is that many enzymes names end in ase. Okay, so for example, amylase. So how do you know that this is an enzyme? Because this word is ending in ase. And the another example of the name of an enzyme is ATPase. Okay, so how do you know that this is the name of an enzyme? Because it is ending in ASE. Well, that cannot be the case in the 100% of the times, but it will be this way in more than 90% of the cases. So this is a good way to recognize the name of an enzyme on your paper. So I have a question for you. So which of the following molecules is an enzyme? A, hemoglobin, B, cellulase, C, monosaccharide, and D, fructose. Anyone?
I think it's B.
B.
Excellent, Isa, excellent, you're good. So how did you get that?
Because it's AS.
Yes, right. So because it is ending with ASE, so that's how you recognize that cellulase must be an enzyme. Okay, excellent. Yeah, that's the correct answer. Okay, so let's proceed and let us discuss more about enzymes, and let's see how do they work. How do actually they speed up a biochemical reaction or a biological reaction within our bodies. So for example, I have added this picture of a charcoal here, and like you, most of you guys must have prepared a barbecue with your families, and this piece of charcoal will not burn if it is like stored in a container, or it is like kept outside in open air. So if you have a piece of charcoal on your hand, will it burn just by itself?
No, no, it
No.
needs a little piece of fire, but to burn.
Yes, yes, excellent. Yes, so it needs heat, it needs energy, right? So it needs energy so that it can start burning. So that energy is called activation energy, and that is the case with everything within our bodies and in chemistry. So everything has an activation energy, and by achieving that activation energy, it starts reacting. And below that activation energy, it does not react. So this is the example of the charcoal, that you need to heat it up before it starts burning and starts reacting with oxygen. So coal is basically carbon, and it burns in the presence of oxygen, and it produces carbon dioxide and water. Okay, so you need heat so that it starts burning. So this energy is called activation energy, and activation energy can be defined as the minimum amount of energy required to perform a reaction or to start a reaction. So we have two graphs here, and in the first one, in graph A, here you can see that this scale is indicating energy, and this scale is representing the progress of a reaction. So this reaction is without an enzyme, and you can see that such a huge amount of activation energy is required before the reactant can be turned into products. Okay, so to change a substrate, or the reactant is also called a substrate. So substrate is anything on which an enzyme acts. So that thing is called a substrate. Okay, so to change a substrate into a product, the energy of the substrate must be briefly raised by an amount known as the activation energy. This can be done by heating the substrate. So what actually enzyme does is that it chemically reduces this activation energy. So here in picture B, you can see that the amount of the activation energy has been decreased as compared to this portion. Okay, so enzyme does not literally heats up the compound or the substrate that it is reacting on inside our bodies, but somehow it chemically lowers the activation energy and it turns it into products more quickly. Okay, so this is how the enzymes work. How do they work? They work by lowering the activation energy of the substrate. So is it clear so far? Any question?
No, it's clear for me.
Okay, so let's proceed. Okay, so now we will see that how actually enzyme recognizes the substrate and convert it into products. So we have basically two theories regarding the mode of action of enzymes. So how do enzymes work? So we have lock and key model hypothesis, and the second hypothesis is induced fit. So we have these two types of theories. So this one, the second one, is considered more fit and more relevant with today's research. And this one is an old model, old theory, but we will discuss it anyway, because this is included in your syllabus. Okay, so lock and key model. So just like a specialized key opens a lock, so similarly, a special substrate can only fit into an enzyme. So lock and key hypothesis, a hypothesis for enzyme action. The substrate is a complementary shape to the active site of the enzyme and fits exactly into the site the enzyme shows specificity for the substrate. Okay, so you can see here, this is a picture of an enzyme molecule, and you can see that it has like a lock-like structure here inside it, and the substrate are resembling the keys. Okay, so this key, which has this round shape, it can fit here, and this another key which has a flat end, it can fit here. Okay, so in our body, like enzyme molecules recognize substrate molecules in a similar way, that they have exact structure for each other, and the site and the position where the keys or the substrate is attached, this is called an active site. Okay, so you can see here that when the substrate is attached, so it forms enzyme substrate complex. The substrate is processed, the activation energy gets lowered, and the product is released. And do keep in mind that during the chemical reactions, the enzymes, they do not get used up. They always stay there after the reaction. Okay, so this is how, according to the lock and key model, the enzyme reactions work within our bodies. So another theory that is the most relevant and the most correct and relevant for to today's research, that is induced fit model. So induced fit model says that the enzyme does not have necessarily to be the exact shape of the substrate. Okay, so you see, this is the active site where the substrate can attach. So you can see that the active site is a little bit different in a complementary way than the substrate than the shape of a substrate, right? So what induced fit model says, that when a substrate comes closer to the enzyme, the active site is changed. The active site is modified according to the shape of the substrate, and it forms an enzyme substrate complex. Okay, so in the previous one, we studied that the enzyme is just like a lock, and it 100% fits a key. But in this model, we are learning that enzyme can a little bit change its shape according to the substrate, and then the product is formed. Okay, so, and this is the most relevant and recent research. And how do scientists came upon this theory, that they scanned the molecules, the structures of the molecules inside our body, and the structure of the enzymes that interact with those molecules. So we have a molecule M, and we have an enzyme E. So they saw that the structure of this molecule, which was reacting with this enzyme, was not exactly similar to the active site of this enzyme. So from there, they realized that, okay, whenever the enzyme and the substrate reactions occur, and they somehow they change a shape. Okay, another example of changing a shape of an enzyme would be, this is also called hand in a glove model. Hand in a glove. So I think I have, okay, so whenever you put on a glove, so you change, like you move your fingers a little bit, and you try to adjust your hand inside the glove. Okay, so the glove is an enzyme, and your hand is a substrate. So hand is also changes its shape a little bit, and the glove is also modified a little bit. Okay, so this is the main difference between these two theories. So any questions for enzymes?
Yeah, I
guys.
have one question.
Please, please,
Is
sure, please
it
ask.
matters it matters the form of the enzyme or not matter what form of the enzyme for the union for union the substrate and the enzyme?
Sorry, your voice is breaking. I couldn't hear you properly. Could you please type or repeat?
I'm gonna type it for you understand
Yes,
me.
yes, 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 please. You type it.
Matthew, send your question by the chat.
Yes. So, Matthew, I will wait for your question. You can type, and in the meanwhile, we will proceed. And when I get your question, I will come back and explain it. Okay, it will save us some time.
It's okay.
Okay, so we are moving on to the next topic, and that is cell membranes and transport. Okay, so in the last lecture, I explained it to you guys that the simple structure of a cell, that it has a nucleus and it has a cell membrane. Okay, so with the help of this cell membrane, things can enter inside the cell and things can exit the cell. Okay, things can come out of the cell and things can get inside the cell. So when something is trying to get inside the cell, it has to cross this cell membrane. Okay, and the same is the case with when something is trying to get out of the cell. So cell needs nutrients, energy, molecules to make energy, to make different molecules, to make DNA, to make proteins. So all the nutrients, the molecules, they want to get inside the cell. Okay, and during the process of making proteins, making DNA, so waste products are created, and the waste products are required to get out of the cell. Okay, so they need to be removed from the cell. So in both cases, they have to cross the cell membrane. So we will discuss the structure of the cell membrane, and we will see that how this transport actually works. Okay, so we have a question here, Matthew. It matters the shape of the enzyme for joining to the substrate, or it don't matter for the union? Well, it does. It does. Enzymes are highly specific, and the shape of the enzyme is very important to recognize a specific molecule. Okay, so in your to answer your question, yes, the shape of the molecule, the enzyme, is very important. But it will there will be a little bit change when it will be attaching with the substrate. Okay, but the overall structure of the enzyme remains constant. That was a very good question, Matthew. Okay, so this is, you can see a picture of a cell wall or a cell membrane. This is the side section of a cell membrane. Okay, the transverse section. I'm showing you a cell wall like this. So we have glycolipids. So these are oil-based molecules. So we are basically discussing the structure of this membrane. Okay, the outer circle. Let me draw it again so that you can see that what we are talking about. So this red membrane, so we are discussing the structure of this red colored membrane. Okay, so we have seen that it contains glycolipids, oil-based molecules. We have carbohydrate part of the glycoprotein. We discussed carbohydrates in the previous lecture. And all these brown, round shaped molecules, they are basically lipid molecules, phospholipids. That is why some healthy oils, fats and oils are necessary in our food. that make up the cell membrane. Okay, just like you eat a meat, if you guys are not vegetarian, so we can take proteins from vegetables as well. So all these molecules are necessary for making different parts of our bodies. And you can see that we have a protein molecule here, a glycoprotein here, cholesterol, phospholipid, and transport protein here. So this is a basic structure of a cell membrane, and this layer is called a lipid bilayer. Okay, bilayer. Bi means two. Okay, and the same word is used, di. Di also means two. Okay, so this is called a bilayer. So this is the first layer, and this one is the second layer. Okay, so let's proceed and let's look a bit more of the structure. So here you can see that this is the phospholipid bilayer of the cell membrane. So we have hydrophilic head. So what do we mean by hydrophilic? Okay, so hydrophilic can be split up into two words, hydro. What do we mean by hydro? Anyone? Aqua, aqua or aqua? Anyone?
Hey guys,
Matthew? Okay, so hydro means water. Okay, and philic, P-H-I-L-I-C. Philic means loving. Okay. So this is a water-loving part of the cell membrane, and that is on the outside. And on the inside, we have hydrophobic regions. So you can see these tails here. So these are hydrophobic tails. So again, hydro means water, and phobic. You may have heard of the word phobia. So phobia means fear. So these molecules, they fear water and they try to stay away from water-based molecules, okay? So we have a hydrophobic tail and a hydrophilic head, and this is a phospholipid bilayer, cholesterol again, some proteins, and the polar region. A polar region is a region that contains a charge, a partial negative or a positive, okay? Just like the poles of the electricity, so we have different poles, right? So in the same way, you can remember that polar region means negative and positive. So you will remember by this example. Okay, so this is the outer surface of the cell membrane. This is the inner surface of the cell membrane, and the transport happens. The transport happens through these molecules, through these proteins that are called the transport proteins, okay? And like by absorption, by osmosis, by diffusion, we will see those in a minute. Okay, so now that we have discussed the structure of the cell wall, of the cell membrane, so now we are going to see the mechanisms by which the transport takes place, okay? So the first one is diffusion. So diffusion is net movement of molecules or ions from a region of a higher concentration to a region of lower concentration down a concentration gradient as a result of the random movement of particles. So can anyone give me an example of diffusion? Anyone? Have you guys studied it before?
I don't study that. Never.
So can you give, okay. Have you studied it or not?
I'm sorry, what did you say?
Have you studied diffusion?
No, I didn't study it.
Okay, okay. So you can do a small experiment at home. They take a glass of water and put a 1 drop of ink, okay? Just 1 drop, fill it with water and put 1 drop of ink in that glass. So after some time, you will see that all this water inside it will the color will change and it will take the shape of the color of the ink. Okay, if you have, for example, if you have inserted red ink here, if you have dropped the red ink, so after some time this whole glass will turn slight red. Okay, so why did this happen? Because of the process of diffusion. Because the water molecules are in constant motion, and they will interact with this red ink, and this red ink will be dispersed, will be spread across this glass of water. Another example of diffusion is like a perfume. Like if you must have a perfume or air freshener in your home, and you can take that perfume and you can spray it in the corner of your room, and after some time you will be able to smell it from the other side of the room. Okay, so the molecules of the perfume, they get dispersed in the air. Okay, so what is happening? So the molecules from the region of a higher concentration, they are going towards the area of a lower concentration, and the molecules of the perfumes are being carried away by the air particles inside our room. Okay, this is a simple way to understand the concept of diffusion. Is it clear?
Yeah, it's all clear.
Okay. Yes, excellent. Leonardo. Leonardo gave us an example. Could be when you cook, the smell travels throughout the living room. Excellent. I'm hungry now. Okay, so now we have another mechanism that helps in the transport, and that is a facilitated diffusion. So the facilitated diffusion, the diffusion of a substance through a transport protein, a channel protein or a carrier protein in a cell membrane. The protein provides hydrophilic areas that allow the molecule or ion to pass through the membrane, which would otherwise be less permeable to it. So here, from this point, the active diffusion takes place, and it requires energy, and the particles get out of the cell, and they also get inside of the cell. And this process is called active diffusion. So another method, or the another process that is required for the transport of through the cell membrane is called osmosis. So these students, these headings are very, very important, and you must know these definitions, and you should learn them by heart. So osmosis, the net diffusion of water molecules from a region of a higher water potential to a region of a lower water potential to a partially permeable membrane. So osmosis is very much similar to diffusion, and the difference is that it has a membrane in between. Okay, so for example, we have lots of students in one small room. Okay, so we have 10 chairs in this room, and we have, let's say, 17 students. So, and here on this side, we have 10 more chairs and only 2 students. So what will the students do? They will try, some of them, they will try to come from this side to this side of the room so that they can have a seat and they can sit down. So here the number of the students is high. On this side, the number of students is low, but it has extra seats. So, but there is a wall in between, and there is a door. So the students will come to this side. So they are using this door to come to this side. During osmosis or during diffusion, we do not have any door, okay? It doesn't matter if we have in diffusion, we do not have any doors. Everything is open in an open area, just like I gave the example of a perfume in a room. So it spreads in the air, but in osmosis, we must have a membrane. This is called a semi-permeable membrane. Semi-permeable means that it allows some things to cross, and sometimes some things are not allowed to cross. Okay, for example, only students can exchange through this door, and you cannot move chairs. So this is how the osmosis work. Okay, so now active transport. So active transport, the movement of molecules or ions through transport proteins across a cell membrane against their concentration gradient using energy, ATP. So during active transport or the bulk transport, so also the energy is required. So bulk transport, we have two types. One is endocytosis, and the second type is exocytosis. These are the examples of a bulk transport. Okay, so the bulk movement of liquids, pinocytosis, or solids, phagocytosis, into the cell by the infolding of the cell surface membrane to form vesicles containing the substance. Endocytosis is an active process requiring ATP. This is the molecule that provides energy, adenine, or adenosine triphosphate. And what is the exocytosis? Exocytosis is the bulk movement of liquids or solids out of the cell. Endo means inside and exo means outside. For example, we have a huge molecule, or like a bacteria or a bacterium, and that is being engulfed by a cell. So this process is called endocytosis. Something is going inside the cell. And exocytosis means getting out of the cells. Exo means out, and endo means in or inside. So I have attached this picture which will elaborate the endocytosis and exocytosis, and it will be 100% clear to you guys. Okay, so look here. This is a bacterium that makes us sick, a germ, and this is a special type of cell that eats this bacterium. Inside our body, we have different types of cells, and nature has placed specialized cells that whenever they discover a bacterium in our body, in tissues or in muscles, for example, you get a cut, a small cut on your skin gets opened, and a germ can get inside, a bacterium can enter. So the nature has a defense system. It has specialized cells like this one, that these cells, they try to eat that bacterium. So that bacterium is is getting inside the cell. So what would we call that? A bacterium entering the cell, endocytosis or exocytosis? Anyone? A bacterium is trying to get inside the cell. What would you call the process?
It will trying to contaminate the body.
Yes, it is trying to contaminate, but the cell is not allowing it, but the cell will eat it. This cell will eat it. This is like a police. It is catching up the bad guys. So now this cell wants to eat the bacterium. So the bacterium will get inside the cell. So what is this process called? Is it endocytosis or exocytosis?
Endocytosis.
How? Why? Why do you think that it is endocytosis? Because endo means inside. bacterium is getting inside the cell, okay? So how does the cell engulf or eat this bacterium? You can see that it has created this cleft within it, and the bacterium came inside here. Now this end and this end, they will be joined, and this end cell will be engulfed. Okay, so this is how the specialized cells that work as a police, they catch the bad guys, they catch the bacterium. Okay, bacterium, or we can say germs. So this bacterium is broken down. So this cell breaks this bacterium, which was trying to make us sick. And now the waste is trying to get out of the cell. So again, this cell membrane will have a small opening, and the waste will leave the cell. Is it clear? And this process will be called exocytosis. Clear?
Sorry, can you hear me?
Yes, I can.
Sorry, I have a micro problem.
No problem. Do you have a question?
No. I at the last time I was trying to answer you, but my microphone are getting fails, and I changed
Okay,
the
no
microphone.
problem. Okay, I can hear you properly now. Okay.
Thank you.
You're welcome. Okay, so this was about the transport, that how things get inside the cell and how they get out of the cell. Okay, so we saw the structure of the cell wall. Okay, and the different types of transport mechanisms that the cell uses to exchange materials in and out of the cell. So now we are moving towards our third topic for today, and that is, guys, very exciting, and that is the nucleic acids and the protein synthesis. Okay, so we are all made up of proteins, like more than 70-80% of our body is made up of proteins. The muscles that you see, the muscles, this is protein. This flesh is made up of protein. Okay, the hair are made up of proteins, and each and everything, most of the things inside our bodies, they contain proteins. Okay, so our teeth, so they are made up of proteins. So we will see that how DNA makes proteins. So this topic is a little bit in detail. So that is why I kept it in the last topic. So we will, let's begin. Okay. So nucleic acids and proteins. So in my last lecture, I discussed that the cell, the nucleus, it contains the nucleus of the cell. It contains DNA, and DNA is a molecule that contains information that what to make inside the cell. Okay, so this is the molecule that controls everything. For example, you have blue eyes. So, and your parents, your mom, your mother also has blue eyes. So how do you have the blue eyes? This DNA was transferred from your mom, from your mother's DNA was transferred to your DNA, and that is why you have blue eyes, just like your mother or just like your dad. Okay. So what is the structure of the DNA? So it has nucleotides. And what is a nucleotide? It consists of 3 parts: a nitrogen containing base. Have you guys heard of acids and bases before? What is an acid and what is a base? Anyone?
No, it doesn't.
Okay. So simply, if I explain a base, it contains a OH group in its formula. And when this OH group is released, so it means that, okay, this is a base. For example, sodium hydroxide, NaOH. So this is a base. So when we will add it into the water, so what will happen? It will split into sodium, Na plus, and OH minus. So this molecule produced this OH, so that is why we call it a base. Okay, so base is a base is any molecule that produces the OH part. Is it clear now?
Yeah, thank you.
Okay, and what is an acid? Acid is something that produces hydrogen ions. So for example, if we say HCl is an acid, so if you mix it in water, what will happen? It will split into H plus and Cl minus, chloride, and this is called hydronium ion, or simply it is called a proton. So this is an acid. Okay, so nucleotides contain nitrogen containing bases and a pentose sugar. Why does it call a pentose sugar? Anyone? You guys from the United States, right?
No, I doesn't.
Okay, so what is a pentose sugar? You may have heard of the building called Pentagon. It's a famous building in the United States. So a pentose sugar, it is called pent, P-E-N-T, because it has 5 corners, 1, 2, 3, 4, and 5. This is why we call it a pentose sugar, and a phosphate group. Okay, so this is the structure of a nucleotide, and the nucleotides together with the nitrogenous bases, they form the backbone of the DNA. So this is a DNA molecule. So you can see you have a pentose sugar here, you have a phosphate, you have a pentose sugar, you have a phosphate, and on this side also you have a pentose sugar, a phosphate, pentose sugar, phosphate, pentose sugar, and a phosphate. And these are the nitrogenous bases. Okay, so this is adenine. We represent this with A, and on this side we have T. So there will always be a pair of A and T. You should remember this, okay? And then we will have cytosine. We have another type of a nitrogenous base, and we have guanine, and we represent it with G. So A and T will always make a pair, and G and C will always make a pair, okay? So here again we have thymine, T, and here we have adenine, A. So you can see that A is always with T, or T is always in front of A, and G will always be in front of C, or C will always be in front of G, or A will always be in front of T. Yeah, that we saw that here in this example. So A, T, T, A, and G, C, or C, G. Okay, so in each and every human being, these pairs are similar, and they make pairs like this: A with T and G with C. Okay, so these are the chemical structures here of the adenine and guanine, thymine and cytosine. So you can see this nitrogen in the structure. That is why they are called nitrogenous bases, because they contain nitrogen atoms. Is it clear so far?
Yeah, it's all clear.
Okay. Okay, so let us move to a very interesting part. So here you can see that the phosphate, this blue portion, is made up of the pentose sugar. And inside this, we have the nitrogenous bases that we just discussed. So again, A is in front of T and C is in front of G, okay? T, A, C, G, G, C, T, A, okay? So I have a question for you guys. T, C, G, A, C, T, A, A. So what should I write here? What will be the complementary for a T? What should come in front of T?
I think
Yes,
that
Matthew.
C.
No, try again.
Let me see. T, C, G, C, T, A, A.
What will come in front of T? I told you in the previous slide.
Oh, T, A.
A, excellent, yes. So this side will be A, and what will come here?
With the C, the G.
Excellent. What will we have here? G.
Z.
C. Now.
With the AT.
Excellent. Yeah, you got it. You got it. Excellent. So you can read and predict the DNA sequence, and that is just amazing. Okay, so I hope the other students are also getting this, and this is very interesting and very, very important for the latest research that is happening these days. So I want you guys to pay attention and try to learn what I'm trying to teach you guys here. So this code, this is a code, right? This is called code. This is called a genetic code. Okay, so this is a genetic code. So we were discussing about proteins. So, okay, I think I will have, I need to restart the computer. The screen just got blanked. I will be back. Stay tuned. Just give me a minute. I need to restart.
Sorry, students. Come back, the teacher Adam right now.
Okay, I apologize. The screen went blank. Okay, so let me share the lecture with you guys, and let's proceed. So we were discussing the genetic code, the DNA, that how we can represent the genetic code. Okay, so I hope you guys can see the screen. Okay, so let us go back to where we were. Okay, so this is a genetic code. Let me share another screen so that to avoid the problem that we just faced. Okay, so now that you know that A will come with in front of T, and G will be paired with C, right? So now let us look at the structure of the proteins. Ma'am, can we have 10 more minutes?
Yes, you can continue.
Okay, just five more minutes, 5 to 10 more minutes.
Ah, it's okay, don't worry, it's okay, you can continue.
Okay, thank you. So proteins, they are made up of amino acids. And there are 20 types of amino acids in our bodies. And their structure looks like a polypeptide. So this is one amino acid. It is connected to another one. It will be connected to another one. And different types of proteins have different numbers of these amino acids. So these are called amino acids. I'm writing AA, amino acid. Okay, so this is the first one, this is the second one, this is the third, fourth, fifth, 5, 6, 7, and 8. So this is a protein molecule. Okay, so this is a simple structure of a protein molecule, and it contains a polypeptide, and these are bonds which connects one amino acid with another one, and they are called disulfide bridges, or these are covalent bonds. Okay, so now how are proteins synthesized from the DNA? Okay, so we have two types of nucleic acids. One is DNA and the other one is RNA. So DNA is a double strand and RNA is a single-stranded molecule. So this table is trying to tell you guys that, for example, this is this is DNA, right? And we have different bases on it. For example, we have A, we have T, we have C, G, C, T, A. So what happens is that this is coded into amino acids. Okay, so 3 of them, so A, T, C, they will code for 1 amino acid. And these 3, G, C, and T, they will code for another amino acid. So in the combination of 3, they make 1 amino acid. Okay, so let me share the slides so that you guys can understand this concept. Okay, so now, so DNA to mRNA and proteins, 5 more minutes. So this is the DNA molecule. It is just like a ladder that you need to climb. Whenever you're trying to climb upstairs, you use a ladder. So it looks like a ladder. So this is a double-stranded molecule, and it splits up. Okay, so this changes into a single-stranded molecule, and it is called mRNA, messenger RNA. And an enzyme comes and you see DNA, RNA polymerase, ASE. So this is an enzyme. So the enzyme will do the work, and it will convert it into messenger RNA, and then the ribosome, this is another part of the cell, it will read the sequence on this single strand of RNA, and it will make it into an amino acid proteins. Okay, so you can see here that this is UAC. So UAC means that, okay, we have to put methionine here. So this will be brought by the transfer RNA. And again, we have AAA, adenine, and adenine. So this will code for lysine. Okay, so methionine will be formed, lysine will be formed, and so on. And here you will have this chain, okay, amino acid chain. And this is how a protein is formed from the DNA. DNA changes into messenger RNA, and then the code is decoded and read by ribosomes, and it leads to a protein. Okay, and just 1 more minute. The DNA is very, very interesting, and just 2 days ago, scientists at the Harvard University, they fixed a DNA. So what are you seeing here? These are 2 rats. Which of them is older and which of them is younger? Final slide, last slide. Anyone? Can you see which of the 2 rats is a younger and which one is old?
I think that the black is the older. The black one is the older.
The black one is younger. Like when people get old, they get white hair. So you can see this white hair.
Oh, I think that the color get more harder with the years.
You know, that's okay. Well try, Matthew. So
I'm sorry.
So this one is old because it has white hair, and this one has black hair, and this rat is young. What if I tell you these two are twins? I have a surprise for you. These two rats were born at the same time, and both of these two rats are of the same age. What happened? So the scientists fixed the DNA of this rat and they turned it back to young age. Okay, so this rat was like this rat few days ago, and they repaired the DNA of this rat and made it younger. Okay, so by fixing the DNA, you can make like animals younger as well and disease free. So this is the latest research that came the two days ago. I hope it is interesting to you. So DNA are very, very important molecules. So now let us summarize what we learned today. Enzymes are biological catalysts made up of proteins. Their name ends in ASE. They work by lowering the activation energy of the reaction. Then we saw the transport, the cellular transport. Cell uses diffusion, osmosis, active transport, and bulk transport for transporting nutrients and wastes through the cell. Then we learned that nucleotides form the backbone of DNA and RNA molecules that contain nitrogenous bases. Always remember G pairs with C and T pairs with A. And at the end, we saw that DNA, the genes, make messenger RNA that makes proteins in a process called translation. So thank you very much. That was all from my side for today's lecture. So if you do not have any questions, so we will, this would be the end of the class.
It's clear, guys. The class is going to be closed right now. Everything is clear?
Yeah, oh, everything is clear for me.
Okay, very good, guys. Excellent class, Aiden. Thank you so much.
Thank you.
Thank you for
Bye.
your participation. Be waiting for the next class, okay?
Yes.
Okay, guys, have a wonderful day. Have a wonderful day, and bye-bye.
Bye.